Good evening, everybody, and welcome. My name's Marina Picciotto. I'm president of the Society for Neuroscience, and welcome to the third Neuroscience 2024 Presidential Special Lecture. As of this afternoon, the annual meeting registration is 22,305. So it's really been a privilege to be part of Neuroscience 2024. I thank all of you for being part of this experience. I hope you've been able to spend some time out looking at posters, looking at all the great work being done by researchers from around the world, maybe even finding your next collaborators. For tonight's PSL, we ask that you silence all your electronic devices now to minimize interruptions. I'd also like to remind you of SFN's photography and recording policy. You can take pictures of the lecturer's work only if they include the little green camera icon. And in the absence of an image or a sign, photography, video, and audio recording and all other forms of recording of the presentation are prohibited. For this talk, you can submit questions at any time during the presentation. Use the Q&A feature in the under the session description on the meeting app or on the virtual platform. The session moderator will ask as many questions as possible, given time, during the Q&A segment at the end of the presentation. And the session recording will be available within 24 hours, and it's going to be accessible on the virtual platform until November 10th. If you do need to leave at any time before the session, please do so quietly. And additionally, as we think of all of the outstanding science that we've gathered to share, it's natural to turn our attention to the next generation of neuroscience leaders, and as such, I'm really thrilled to recognize two outstanding cohorts of trainees, participants in the Neuroscience Scholars Program and the Early Career Policy Ambassadors Program. You all know that in an increasingly global and diverse field, SFN advances brain science by serving as a professional hub for neuroscientists, providing scientific training, professional development, advocacy, and career opportunities, as well as this opportunity to foster an international community of colleagues. As part of those initiatives, the Neuroscience Scholars Program, also known as NSP, is a 2-year in-person and online training program to support graduate students and postdoctoral researchers from underrepresented populations, those living with disabilities, or those from disadvantaged backgrounds. NSP is more than 40 years old, and it's funded by a recently renewed grant from NINDS, and it's successfully supported an incredible group of neuroscientists through its programming and mentoring. The NSP alumni network is strong, and we're excited to welcome the new cohort of more than 200 scholars to the group. The success of this field requires the inclusion of all our voices to fuel excellence in science. For those in the Early Career Policy Ambassadors Program, your interest and commitment in advocating for the field of neuroscience is incredibly valuable and necessary, from raising awareness among your fellow neuroscientists all the way to discussing the importance of continued support for neuroscience with your federal legislators. The work you're doing has already and will continue to make a difference for our community, and we commend you for your efforts. Your involvement with the Society lead is an essential contribution to the field of neuroscience and continues the growth of a society dedicated to advancing the field throughout your careers. So let's first thank them. And now, I would like to invite all of the NSP and ECPA participants, past and present, to stand and be recognized. So before welcoming tonight's lecturer, we will present 4 prestigious awards recognizing a range of accomplishments from early career achievements to career-long successes to contributions to neuroscience education and training. The Bernice Grafstein Award for Outstanding Accomplishments in Mentoring recognizes individuals who are dedicated to promoting women's advancement in the field of neuroscience and who've made outstanding accomplishments in mentoring. This award is made possible by the generous support of Dr. Bernice Grafstein, a distinguished neuroscientist and a past president of SFN. The recipients of this year's award are Dr. Nina Shore and Dr. Marina Wolf. Dr. Shore is the Deputy Director for Intramural Research in the National Institutes of Health's Office of the Director and serves as Neurology Director for the American Board of Psychiatry and Neurology. During her distinguished career as a pediatric neurologist, Dr. Shore's research focused on neural crest development, neuronal cell death, and targeted therapy for neuroblastomas. She's received numerous commendations for her teaching and mentoring, and she has mentored more than 80 students and postdoctoral fellows, as well as numerous medical students and early career faculty. In addition to her leadership roles at NIH in strategic planning and career development programs, Dr. Shore's played a key role in creating national programs that have empowered countless female neuroscientists. So I would like to present this to Dr. Shore. Dr. Wolf is a professor of behavioral neuroscience at Oregon Health and Sciences University and a leading researcher in the neurobiology of substance use disorders. Her name is also Marina. Her research is focused on the role of synaptic plasticity in animal models of drug addiction, and it has implications for treating substance use disorders in humans. She's known for her staunch support of students and postdoctoral fellows in her laboratory. And Dr. Wolf is also dedicated to inspiring women to consider careers in neuroscience and to mentoring neuroscientists so that they remain in the field. Through membership in professional societies, including SFN, Wolf has advocated for diversity, including professional development programs for female scientists, and she's led such efforts as a past president of the American College of Neuropsychopharmacology. Dr. Wolf? The Janet Rosenberg Trubach Career Development Award, generously supported by the Trubach family, honors 2 mid-career individuals who've demonstrated originality and creativity in research as they continue to develop as neuroscientists. The award is named after the late Janet Rosenberg Trubach. The 2 recipients this year are Dr. Amber Al-Hadef and Dr. Vijaya Rangaraju. Dr. Al-Hadef is an assistant member at the Monell Chemical Senses Center and adjunct assistant professor in the Department of Neuroscience at the University of Pennsylvania. Her research examines how external stimuli such as food and drugs influence neuronal activity in specific brain circuits in mice. Her work combines sophisticated in vivo techniques with advanced microscopy, behavioral, and surgical approaches to examine the neural activity underlying gut-brain signaling. Her latest groundbreaking research is the discovery that separate neural circuits mediate the weight loss effects versus aversive side effects of GLP-1 receptor agonists. Congratulations, Dr. Al-Hadef. Dr. Rangaraju is a research group leader at the Max Planck Florida Institute for Neuroscience. Her work explores how the energy created by mitochondria influences neurons involved in learning, memory, and behavior. As a PhD student, she developed sophisticated ways to visualize ATP levels inside nerve terminals during synaptic transmission. This resulting sensor has been patented to measure ATP in cells. Now, as principal investigator of her own laboratory, she's identified key proteins and dendrites that tether mitochondria near synapses to support learning and memory. Her continued research into mitochondrial stabilization and local energy supply in neurons has direct implications for the disruptions in motor and cognitive function in neurodegenerative diseases. Congratulations, Dr. Rangaraju. The Louise Hanson Marshall Special Recognition Award honors individuals who have significantly promoted the professional development of women in neuroscience through teaching, organizational leadership, public advocacy, or other efforts. The two recipients this year are Dr. Laura Andre and Dr. Michelle Jones-London. Dr. Andre is a reader at the Center for Neurodevelopmental Disorders at King's College London and an accomplished researcher known for her work on circuit formation in neurodevelopmental disorders. She was pivotal in establishing infrastructure to promote diversity, equity, and inclusion in neuroscience internationally. Upon joining the ALBA network in 2020, she helped develop the ALBA Declaration of Equity and Inclusion, which provides evidence-based guidelines for individuals and organizations. She's been equally active within the United Kingdom, where she was instrumental in creating the British Neuroscience Association's Scholars Program, which provides mentorship, networking opportunities, and financial support to students and early career researchers from underrepresented ethnic groups. I want to congratulate Dr. Andre. Dr. Jones-London is the Associate Director of the Office of Programs to Enhance the Neuroscience Workforce, also known as OPEN, in the National Institute of Neurological Disorders and Stroke Office of the Director. At NINDS, her work comprises three complementary yet distinct domains: improving neuroscience research and neurological health across the country via training and development programs, using evidence-based research to create inclusive communities of researchers, and promoting welcoming environments for the entire NINDS workforce to advance the Institute's mission. Dr. Jones-London designed more than 5 programmatic interventions to increase diversity and inclusion at NINDS and across NIH. And her visionary leadership and her ability to motivate diverse collaborators successfully have resulted in measurable DEI advances in the NIH neuroscience and the biomedical workforce. I also want to mention that Dr. Jones-London is an NSP alumna. Congratulations, Dr. Jones-London. Our final award of this evening is the Mika Salpeter Lifetime Achievement Award. which was established in 2000 in celebration of the 20-year anniversary of the Women in Neuroscience organization, and it's now presented on behalf of the Inclusion and Diversity Programs Subcommittee of SFN's Professional Development Committee. Each year, the award recognizes an individual with outstanding career achievements in neuroscience who has also significantly promoted the professional advancement of women in our field. SFN remains deeply committed to its core value of supporting, engaging, and welcoming diverse voices and scientific exchange between scientists of all nationalities. This year's recipient is Dr. Maria Grazia Spillantini. Dr. Spillantini is a professor at the University of Cambridge in the Department of Clinical Neurosciences. Her work has greatly contributed to the understanding of neurodegenerative diseases and how tau and alpha-synuclein form aggregates in neurons. Her pioneering research established that tau dysfunction is sufficient to cause neurodegeneration and dementia in disorders such as Alzheimer's disease and frontotemporal dementia, and that alpha-synuclein dysfunction is linked to Parkinson's disease, Lewy body dementia, and multiple system atrophy. She's published more than 300 scientific articles. She is a Fellow of the Royal Society and the UK Academy of Medical Sciences. She's won numerous awards, including the Potemkin Prize, and she's been made Knight Officer of the Star of Italy by the President of the Italian Republic. That's pretty cool. Dr. Spillantini is a vocal advocate for greater gender equality and diversity in neuroscience and champions women in science through empowerment, sponsorship, and mentorship. And her former trainees say that they felt prioritized above all. Congratulations, Dr. Spillantini. And now I'm really pleased to introduce this evening's presidential lecturer, Dr. Nancy Ip. Dr. Ip is the president of the Hong Kong University of Science and Technology and the Morningside Professor of Life Science. She is the first female president of a publicly funded university in Hong Kong. Dr. Ip received her PhD in pharmacology from Harvard University. She's well known for her outstanding work in the field of neuroscience and has contributed to the understanding of complex mechanisms that underlie proper brain function, as well as neurodegenerative disease treatment discoveries. Her talk tonight is titled, Unveiling the Nexus of Innate Immunity and Alzheimer's Disease, Insights into Pathogenesis and Therapeutic Prospects. Please join me in welcoming Dr. Ip to the stage.
Thank you, Marina, for your generous introduction. It is really a great honor to be here to give the Presidential Special Lecture. I deeply appreciate the opportunity to share with you the latest findings from my lab on innate immunity and Alzheimer's disease. Well, Society for Neuroscience always have a very special place in my heart. Since I became SFN member more than 4 decades ago when I was a graduate student. It is truly a remarkable organization. And being at this podium today reminded me of the special lecture that I gave 19 years ago in Washington, D.C., where I talk about the role of a kinase in synapse development and plasticity, which forms the cellular and molecular basis for memory formation. So today, I'd like to talk about some of the highlights of our research findings in the past decade as part of our efforts to understand the biology of Alzheimer's disease. This is my disclosure statement. I'm sure you agree that Alzheimer's disease, AD, is an urgent health threat globally. With the aging population worldwide, there's an increase in the prevalence of age-related neurodegenerative diseases, such as AD. So AD is the most common form of dementia, and it is projected that there will be more than 100 million AD cases by 2050, causing a huge economic burden on society. So the discovery of AD was more than a century ago, when Dr. Alzheimer described this disease as a peculiar disease of the cerebral cortex. The clinical symptoms include progressive memory loss, impaired memory and impaired reasoning and judgment, as well as behavioral and psychological symptoms. Now, the clinical symptoms appear in moderate to advanced stages of the disease. However, there are already pathological changes in the brain 10 to 20 years before the onset of the clinical symptoms. The hallmarks of AD include extracellular amyloid plaques comprising amyloid beta, A-beta, and intracellular neurofibrillary tangles comprising hyperphosphorylated tau protein. Now, A-beta clearance is mainly mediated by microglia, which are the primary innate immune cells of the brain, and rapidly respond to injury, pathogens, and abnormal protein aggregates such as A-beta in order to maintain brain homeostasis. Well, early histological studies reveal that there is close association of microglia with AD pathologies. Microglia undergo morphological changes in the AD brains. Microglia aggregates around AD brain lesions, such as the amyloid plaques. And microglia express on the cell surface proteins that are indicative of phagocytic function, such as MHC class II protein. Now, our understanding on the role of innate immunity in AD have been enhanced by genetic studies over the last few decades. So since 1990s, investigation of the genetic underpinnings of AD serves as a powerful approach for us to elucidate the biological pathways that underlie AD pathogenesis. So some of the earliest genetic determinants that were discovered include amyloid precursor protein, presenilin 1, presenilin 2. So these are the genes that accounts for the autosomal dominant AD. So such findings really reveal the pathogenic role of amyloidogenic APP processing. And around the same time, there was discovery of ApoE4 as a strong risk gene of late onset sporadic AD. And this finding implicates the role of lipid homeostasis. Subsequently, in the year 2000s, there was a large-scale twin study showing that 60 to 80% of AD risk can be attributed to heritable factors. And around that time also, there was a lot of genome-wide association studies, GWAS, that identified numerous AD risk genes implicating innate immunity. And CD33 was identified as the first AD risk gene that were found on microglia. And TREM2 variants was also identified around the same time. Now, GWAS have linked many innate immune genes with AD, such as ApoE and TREM2. And interestingly, AD susceptibility loci are enriched in microglial promoters and enhancers. So this regulation in the levels of microglial genes may impact the pathology and the progression of AD, and all these suggest that microglia may play a causal role in AD pathogenesis. The deposition of amyloid around neurons results in neurotoxicity, and the accumulation of tau and the hyperphosphorylation of tau leads to neuronal death. So for microglia, they sense and respond to disease, to damage-associated molecular patterns, DAMPs. And these can include A-beta, apoptotic neurons, dystrophic synapses, and myelin debris. So in response to these damage-associated molecular patterns, microglia engage in 3 major functional pathways in order to mitigate damage and exert neuroprotection. The first one is for microglia to sense and to migrate towards the sources of DAMPs, to engulf and degrade DAMPs. Second, the microglia form a barrier around the amyloid plaques, compacting them and also minimizing the exposure of neurons to DAMPs to reduce neurotoxicity. Thirdly, microglia can also release soluble mediators, and these can include neurotrophic factors, chemokines, and cytokines to facilitate the resolution of immune response and promote tissue repair. However, upon chronic exposure to DAMPs, the unresolved inflammation will lead to detrimental damages in the brain. So there are 4 steps in the clearance of the DAMPs. First, microglia will recognize and migrate towards DAMPs, engulf them, and digest them through endolysosomal processing to smaller components. Then microglia will undergo an adaptation phase and can undergo functional state changes. Now, there are many AD risk genes being identified, and they play major roles in a microglial clearance pathway. And examples are shown on this slide, for example, including ApoE and TREM2. Our understanding on the roles of genetic risk factors in AD pathogenesis are really enhanced by the development of various experimental approaches. So histological studies together with omics data provide critical insights into how AD risk factors can affect the molecular and cellular phenotypes of disease, and development of various mouse experimental models, including the AD transgenic mice, the knock-in mouse models. These actually would enable the mechanistic studies of genetic variants of risk genes in AD in vivo. And thirdly, the generation of brain cells from human iPSC would enable the studies of how genetic variants can impact specific brain cells in the context of the human genome. So it is important for us to delineate the molecular pathways that can mediate the microglial response to AD pathology. Now, using advanced technologies, several labs, including ours, we use single-cell, single-nucleus transcriptome profiling, and we uncover a heterogeneous subpopulation of microglia. So microglia exhibit multiple cellular and functional states in response to environmental stimuli. Trajectory analysis allows us to predict the cell differentiation path using single-cell RNA sequencing. So in AD, the homeostatic microglia can transit to distinct functional states, such as inflammatory, chemotactic, and phagocytic. So we would like to investigate the special function of microglial subpopulations and their state transition, because it would allow us to understand better the roles of microglia in AD pathogenesis. So along the disease progression, the microglia actually express a lot of cell surface receptors that would allow them to sense the DAMPs and to respond to it. Other cells, including glial cells and neurons, they also would release different cytokines, such as inflammatory cytokines, such as chemokines, and complement factors, as well as components. Now, the signaling axis that I would like to share with you today is the IL-33 ST2 signaling. And this is a pathway that my lab started to work on about 8 or 9 years ago. So interleukin-33, IL-33, is a member of the interleukin-1 receptor family. It acts as an alarmin, and it is rapidly released upon cellular damage in order to maintain the immune homeostasis. IL-33 binds to a heterodimeric complex that is comprised of ST2 and IL-1 receptor accessory protein. And there are links between the IL-33 ST2 signaling and AD. For example, genetic variants of IL-33 and also ST2, they are associated with AD. Another finding is that IL-33 gene expression is decreased in AD patient brains. So with these observations, we embark on a study to look at the outcome of administering IL-33 to APP-PS1 mice. So Nelson and Amy, they injected IL-33 into this AD transgenic mice. And we were very excited to observe that the amyloid plaque pathology is much reduced following administration of IL-33. And the quantitation is shown here. In addition to reducing the A-beta pathology, IL-33 administration also ameliorates the hippocampal synaptic plasticity impairment, as well as alleviating the memory deficit. So given that ST2, that is one of the heterodimeric receptor complex of IL-33, they are expressed on microglia. So we ask if microglia are the mediators of the beneficial effect of IL-33 that we observed. So we injected IL-33 into the APP-PS1 mice. And as we observed before, the A-beta plaque is much reduced following IL-33 treatment. We also noted that IBA1 positive microglia, they are located in the core of the amyloid plaques. So using 3D reconstruction, we look at the microglia that are co-localized with A-beta, and we found that IL-33 administration can increase this co-localization. And again, we would like to know if these microglia, they have enhanced phagocytic activity. So we use the cell surface marker, CD11b and CD45, to look at these resident microglia, and we use methoxyXO4, the A-beta labeling dye, to look at the ability of these microglia to take up A-beta. So at the resting stage, before IL-33 administration, roughly 10% of the resident microglia will uptake the A-beta. But following IL-33,
it enhances the percentage of cells that take up A-beta. Now, for these microglial population that respond to IL-33, we found that IL-33 induces the MHC2 expression for these A-beta associated microglia. So for these experiments, again, we look at the ability of IL-33 to induce this microglial subpopulation that show expression of MHC2 protein. So again, this is part of the antigen presentation complex. And this microglial subpopulation that respond to IL-33 also show enhanced ability to take up A-beta. Again, this is done using flow cytometry analysis. So the MHC2 expressing phagocytic IL-33 responsive microglia, they are the major cellular targets that mediate the IL-33 stimulated beneficial functions of microglia. So we perform in vivo imaging experiment to really track the phenotypic changes that occur. And for that purpose, we generated the APP-PS1 63CL1 EYFP mice. So for this particular mouse model, the microglia expressing EYFP is shown here in green. So under controlled conditions, you can see that the microglia seems to be rather stable and not much movement. However, after we treat the mice with IL-33, you can see that some of the microglia start to migrate towards the amyloid plaque as depicted in red. And the quantitation on the right-hand side here show that the distance of the A-beta to the microglia actually is reduced over time. So IL-33 treatment induces microglial subpopulation with specific functional characteristics in the AD transgenic mice. We observed different phenotypes. So at 3 to 8 hours, the microglia start to migrate towards the A-beta plaque, and at 24 hours, they will show phagocytic activity. So we would like to investigate what are the molecular signatures of these microglia that have the specific functional characteristics during IL-33 stimulated A-beta clearance. So Kevin and Walter, they conducted this study using the IL-33 treated APP-PS1 microglia and performed the single-cell RNA sequencing analysis. So they identified 3 subpopulations of microglia: homeostatic, chemotactic, and disease-associated microglia. And we noted that the profile actually shifted over time. So a homeostatic subpopulation will be decreased, and the chemotactic subpopulation will increase over time during 3 to 8 hours after IL-33 treatment. Now, the trajectory and pseudotime analysis can be used to predict the state transition of microglia. This experiment is again done with AD transgenic mice and with IL-33 treatment. So cell trajectory and pseudotime analysis show that the chemotactic state, that is the yellow part, is actually an intermediate state in the IL-33 responsive microglia developmental lineage. And you can see on the heat map on the right side that there are chemotactic gene expression that is induced during this intermediate stage. So how do the microglia sense and then move towards the amyloid plaque? To conduct this study, Kevin and Walter, they identify the genes that are increased in a chemotactic microglia. So out of the 142 genes identified, 39 of them represent the cell surface protein. And we found that out of the 39, 3 are the candidate surface receptors that can potentially mediate IL-33-induced microglia chemotaxis towards the amyloid plaques. So which one of these 3 is the responsible candidate surface receptor? So I just want to give you some examples of the experiments that were done. So one approach was to use the blocking antibody treatment for the candidate receptors. So these blocking antibody was administered to these mice using ICV delivery, and this was done before IL-33 treatment. So we found that the blocking antibody against VCAM1, as shown, the quantitation is shown here. This is the candidate cell surface receptor that can abolish IL-33-induced microglial chemotaxis towards amyloid plaques. The other candidates, ICAM1 or CCR7, the blocking antibody had no effect on this phenotype. We also use another approach where we generate the microglial conditional knockout of VCAM1 in APP-PS1 mice. So if we genetically ablate the microglial VCAM1, it can abolish the IL-33-induced A-beta clearance in the AD mouse model. So here, in the wild-type VCAM1, APP-PS1, you see the MHC2 positive microglia, but then when we knock out VCAM1 using the induced conditional knockout approach, this is abolished. We also observe that the amyloid plaque load is also abolished. So normally it can be reduced, but in the knockout, you can no longer see the reduction of the amyloid plaque load. So this is, again, another supportive evidence that VCAM1 is the cell surface receptor on microglia that is mediating chemotaxis. But how do these microglia then sense the chemoattractant in a A-beta plaque so that they can direct the migration towards the plaque? Now, using mass spec, there are about 20 A-beta plaque-associated proteins identified. And out of these 20, when we use the protein-protein interaction analysis, there are 4 A-beta plaque-associated proteins that can interact with VCAM1. So which one of these 4? Well, for these experiments, what we did was we coat the beads with different proteins, so ApoE, CD44, or ITGB2. And we found that when we inject these beads into the brain of the mice, and then we look at the migration of microglia towards these protein-coated beads, only the beads coated with ApoE were we able to trigger the microglial chemotaxis in the IL-33-treated APP-PS1 mice. The other 2 did not have any effect. We also use another approach where we block the ApoE signaling using a neutralizing antibody, and this is done in collaboration with David Hossman. And when we treat the mice with a blocking antibody against ApoE, we found that the MHC2 positive microglia that is characteristic of the response to IL-33, this MHC2 positive microglial subpopulation is totally abolished when we administer the neutralizing antibody for ApoE, and the reduction of the A-beta plaque was also abolished by the neutralizing antibody against ApoE. So we know that actually distinct microglial subpopulation, they express different specific genetic signatures. For example, I just shared with you that the phagocytic IL-33 responsive microglia, they have the distinct MHC2 gene signature. The transcriptomic regulation of microglia is regulated by epigenetic remodeling. So we want to examine how IL-33 can regulate the epigenetic landscape in microglia. And for these experiments, we use a TAC-seq analysis. We found that there is increased chromatin accessibility at the promoter regions of the MHC2 genes in IL-33 responsive microglia. And when we look at the motif, after performing the analysis, we found that the chromatin accessible regions are enriched with the PU1 transcription factor motif. So when the chromatin accessibility is increased, there is increase in the binding of transcription factor, and we believe that PU1 is such a transcription factor that bind to that region. So IL-33 increases the chromatin accessibility of the regulatory region of MHC2 gene in that microglial subpopulation which respond to IL-33, and that enhances the binding to PU1. So we perform a lot of experiments to convince ourselves that, in fact, PU1 is the transcription factor that regulates the overall transcription. And one experiment that we did is to use the inhibitor against PU1, DB2313. And we found that administration of DB2313 can abolish the effect of IL-33. For example, we look at MHC2 positive microglia in response to IL-33. We found that DB2313 can totally abolish that. And this PU1 inhibitor can also abolish the characteristic reduction of A-beta plaque in the presence of the inhibitor. So I just want to summarize this part of the talk where I show you that IL-33 can stimulate the induction of the chemotactic IL-33 responsive microglia and the migration towards the plaque. And this process is mediated by the cell surface protein VCAM1, which can sense the A-beta plaque-associated ApoE in order to bring about this chemotaxis. So the IL-33 responsive microglia adopt an MHC2 positive phagocytic state for A-beta clearance, and PU1 is the transcription factor that can mediate this effect of IL-33. So the IL-33 ST2 signaling that I described to you seems to have a beneficial effect using the AD transgenic model mice. Is this signaling dysregulated in AD? Now, I mentioned that IL-33 act on a heterodimeric complex, ST2 and IL-1 receptor accessory protein. It turns out that ST2 can also exist in a soluble form called soluble ST2, and it acts as a decoy receptor to inhibit the IL-33 signaling. Now, Jason and Fred, they look at the level of soluble ST2 in the plasma in healthy control as well as AD patients. And they noted that there's an elevated level of soluble ST2 in the plasma. There's also an elevated level of soluble ST2 in the cerebrospinal fluid of those patients. And we found that the soluble ST2 level positively correlate with the progression of amyloid deposition in the AD brains. So we asked the question, what is the outcome of injecting soluble ST2 into the AD transgenic mice? We performed the injection using ICV approach, intracerebral ventricular injection, and we found that infusion of soluble ST2 can exacerbate the A-beta plaque load, as shown here. And we also noted that injection of soluble ST2 can reduce the microglia that exhibit the A-beta uptake. So from these data, it shows that soluble ST2 has a negative effect in terms of exacerbating the AD phenotype. So we want to understand more about the regulation of soluble ST2. So we perform whole genome sequencing and GWAS analysis in a patient cohort with soluble ST2 data. So we found that the variants at a particular locus called IL1R, L1 locus, they are associated with the soluble ST2 level. Now this locus encodes both the regular ST2 form and also the soluble ST2. We also found that carriers of this particular allele that we identify called, this is too long for me to call. So this is an allele of the soluble ST2, and they seem to, for individuals that carry this particular allele, they have lower soluble ST2 level, as shown here, both in the plasma and also in the CSF. So we analyzed whether there is any implication in terms of the AD risk. We found that for female ApoE4 patients with AD who carry this particular genetic variant, they have a lower level of soluble ST2 in the plasma. They also exhibit a lower risk of AD by about 20 to 40%. So this indicates that this particular genetic variant is a protective variant. So we went on to look at where is the source of this particular gene expression. So we conducted a single nucleus RNA-seq analysis using postmortem human brains obtained from the UK Brain Bank. We found that the soluble ST2 gene is mainly expressed in the endothelial cells, as indicated by the marker CLDN5 in the human frontal cortex. We also found that this particular variant is associated with reduced soluble ST2 expression. So there seems to be a cell type specific effect on the soluble ST2 gene expression. So we then asked the question, if we delete the genomic region that harbor this particular locus, will it decrease soluble ST2 expression and secretion? So we did this experiment using the CRISPR-Cas9 approach, and we successfully removed the genomic region that harbor the genetic variant. As shown here, both the soluble ST2 expression and the soluble ST2 secretion, they were abolished when we removed this genomic region that harbor the genetic variant. So deleting that region can abolish the expression and also the secretion, showing that, in fact, this particular locus is a key genetic factor that regulates the soluble ST2 level. Then we look at the consequence. If you have lower soluble ST2 level, what would happen to the AD-related endophenotype? We found that for the carriers of this protective variant, there's a delay in the age of AD onset. We also found that there's an increase in the brain region volumes, in particular the entorhinal cortex. So this particular region of the brain is one of the earliest brain region that seems to have dysfunction and show the A-beta, the amyloid plaque phenotype. We validated this result using yet another cohort obtained in Australia. This is in collaboration with Colin Masters. We found that for the carrier of this particular protective variant, the AA variant, the gray matter volume atrophy is actually reduced compared to the non-carrier. So this further confirmed that lower soluble ST2 level have a protective effect against brain atrophy in the brain. So to provide further information that lower soluble ST2 level is indeed beneficial in terms of reducing A-beta deposition in patients with AD, we use postmortem human brain sections, and we look at the A-beta plaque load. You can see that comparing the carriers of this particular protective variant versus the non-carrier, the protective variant carrier show that there is a more significant reduction in A-beta plaque load. We also show that there's more interaction between the microglia and the A-beta. So again, the quantitation is shown here. There seems to be more interaction between the A-beta and the microglia that migrate towards the A-beta. Lower soluble ST2 level is also associated with increased microglial response. So as shown here, we perform association tests between the soluble ST2 variant and the microglial gene expression in AD. And we found that there is an upregulation of A-beta responsive genes, which we know that would lead to enhanced A-beta uptake and degradation. So the molecular signatures is consistent with what we observe in terms of the endophenotype. Now, if we correlate the molecular phenotype of microglia with the spatial information, then it can provide insights into the interaction between A-beta and the microglial subpopulation that show distinct function. So what I show here is that when we identify the homeostatic microglia using this particular marker, this cell is right here, or we identify the A-beta responsive microglia using the marker that can identify MHC2, we found that these two microglial subpopulation, they exhibit different localization with respect to the microglia. That is, the homeostatic microglia seems to be away from the A-beta plaque, which is in purple, whereas the A-beta responsive microglia here, they are interacting or at least near the A-beta plaques. So this kind of study on the spatial localization can provide information on the cellular heterogeneity of microglia and also provide information on how they might react to changes in the microenvironment. So far, I have described to you that IL-33 normally exert a beneficial effect via the microglia. But when soluble ST2 is enhanced, it can act as an inhibitor of the IL-33 ST2 signaling, and it would result in increase in A-beta deposition. I also reported to you that we have identified a genetic variant that can decrease the soluble ST2 expression, in turn enhance the responsiveness of microglia to A-beta, decrease A-beta pathology, and is a protective variant against the AD risk. So we believe that targeting soluble ST2 may be a viable approach for therapeutic development. And we had 2 posters this afternoon on soluble ST2 and another blood protein, soluble TREM1. So the proof of concept using the IL-33 ST2 signaling pathway help us to dissect the biological functions of genetic risk variants of AD and elucidate novel pathological mechanism. So we believe that the approach of functional genomics can help us to investigate the relationship of genetic variants and risk genes with a particular disease in question, and in this case is AD. So genetic risk factors, such as the one that I just mentioned, and the effect on the phenotypes, for example, the soluble ST2 level, and the pathological pathways involved, that is, impairment of the microglial A-beta clearance. I think all these studies will help us to understand better the pathophysiology of AD and help us uncover promising therapeutic targets, such as soluble ST2. But I believe that it is important for us to have better representation of other ethnic populations besides the European descent, because these other populations, they are less than 10% of the total AD genetic studies. And we know that the genetic associations with AD risk can differ between populations of European descent and other populations. Prime examples are ApoE. The risk effects of ApoE4 vary among ethnic groups. And also for TREM2, the prevalence of the risk mutations, they are also different between European and the East Asian population. And so with this in mind, we work with John Hardy to build this in-house AD cohort, and the work was mainly done by Fred, Chen Yu, and Fanny in collecting these samples. So using this database of the Chinese population, we identify novel genetic variants in immune-related genes. We identify risk-associated ApoE haplotypes in Chinese population. We identify common variants of soluble ST2, just as what I show you. And we also identify TREM2 H157 variant carriers, and this is the topic that I want to share with you next. So TREM2, of course, is a very well-known risk gene, and it plays a very important role in mediating microglial response to AD pathology. Now, multiple risk variants of TREM2 have been identified, and they are mainly located at the ligand binding region. And for these variants, there is reduction in ligand binding affinity. There's also attenuation of the TREM2 downstream signaling. In addition, the loss of TREM2 can compromise the microglial protective response against amyloid pathology, including compromising the barrier formation. So there is another variant that I mentioned earlier, H157Y. And a single copy of this variant can lead to substantial increase in the risk of AD with an odds ratio of more than 6. And this particular variant is associated with AD in the Chinese population. This is a work that I would like to share with you next. There's a 5-fold increase in the allele frequency in the Chinese population. And interestingly, for this particular variant, it is not at the ligand binding site, but nonetheless is external to the cell surface. And hence, upon proteolytic cleavage, there's an increase in soluble TREM2, and it can also affect the TREM2 downstream signaling. So we look at the effect of the genetic variant on AD pathological outcome and the progression. And the work was done by Jackie and Dennis. So they found that this particular variant is associated with more severe disease outcome and more pronounced neurodegeneration. And these are brand new data, not published. So they found that if they compare the carriers of this variant with the non-carriers, they have increased brain atrophy, and they also, for the AD patients, they have increase in the brain atrophy. And very interestingly, for the MCI patients, which stand for mild cognitive impairment, and these patients have a high risk to progress to AD. For these individuals, they observe increase in the volume loss in the entorhinal cortex. So these, again, they are not AD patients, they are MCI individuals. And they use blood-based biomarkers to look at the association of this variant with AD pathology and neurodegeneration. Again, the groupings are cognitively normal, mild cognitively impaired, and AD. And using the blood-based biomarkers such as phospho-tau 217, which is an AD biomarker, and also neurofilament protein, a very good indicator of neurodegeneration. They found that for the TREM2 variant carriers, they have more significant rapid neurodegeneration shown here in AD patients and shown here in the mild cognitive impaired individuals. So again, very recent data, like just a few weeks ago, when they conducted the blood protein profiling of the cognitively normals, that is not MCI, not AD, but cognitively normal. These individuals, if they carry the TREM2 genetic variant, they already show alteration of immune-related pathways. So we believe that these findings are very important to understand better the AD pathogenesis. So blood protein profiling indicates the biological state that associates with genetics and disease. So our lab was one of the very first labs to conduct the comprehensive proteome profiling a few years ago. And we screened more than 1,100 blood proteins in control, MCI, and AD individuals. So after we look at the protein levels, more than 1,100 of these proteins, we identified more than 400 of these blood proteins that are altered in MCI and AD. So we believe that the blood protein biomarkers that we identify can represent the blood protein signature of patients with AD or MCI. So the way we did the analysis is that we perform co-regulation and network analysis, and we clustered the proteins into different groups. And each cluster is associated with distinct biological processes, and they also exhibit unique cell type expression profile. And for each cluster, we identify one protein, and we call it the hub protein. So this will allow us to have a more comprehensive evaluation of AD status. It also facilitates early screening and staging of the disease. So the blood proteins, we believe, indicate early innate immune dysregulation in AD. Now, we can group them into three types. One is early dysregulated proteins, where you can see the change in the MCI individuals. Another one is the other side of the spectrum, where you only see the change in the AD patients. So these are the AD stage proteins. So what we found intriguingly is that the innate immune response changes can be observed early on, but the inflammatory response changes are observed later on. So along AD progression, there is an early downregulation of innate immune proteins and late upregulation of inflammatory proteins. So we believe that the blood proteome can reveal multi-pathway analysis of the AD status. So we then have 5 groups, 5 different biological pathways, including neurodegeneration, inflammation, innate immunity, vascular function, and metabolic activities. So we can evaluate multiple biological processes at the same time that are involved in AD, and we can achieve a multidimensional and a comprehensive analysis of AD status. So using a multi-scale scoring system for assessing the activities of these 5 AD-associated biological processes, we can have distinct dysregulated patterns in MCI and AD. So for example, innate immunity is dysregulated in MCI patients that have A-beta positive PET scan, not in A-beta negative PET scan. So this is a very significant finding, we believe. And it's also dysregulated in patients with AD, of course. So the innate immunity is one pathway that is dysregulated in these 2 groups. Well, we are, of course, very excited that there are 2 FDA-approved drugs for AD using monoclonal A-beta antibodies, and they are lecanemab and donanemab. And these 2 drugs, 2 antibodies, so they can trigger microglial response against amyloid plaques. So they trigger this response, and then the microglia will uptake and clear the A-beta. So I believe it is timely for us to consider the approaches of promoting protective response and restoring the normal functions of microglia. So for ApoE4, there are ongoing efforts to modulate ApoE functions. For the TREM2 variants that lead to loss of function of TREM2, there are ongoing efforts to activate TREM2 using the agonistic antibodies. And for soluble ST2 that I shared with you earlier, the genetic variant that can reduce soluble ST2 level in a plasma, this suppression of soluble ST2 can enhance IL-33 signaling and also enhance microglial chemotaxis and phagocytosis. We believe that soluble ST2 is a good target, so we already went ahead to try ways to modulate the soluble ST2 level. And I just want to share with you one proof of concept experiment where we generated knockout of soluble ST2 in the AD transgenic mice. And we found that knocking out soluble ST2, but not the full-length ST2, we were able to show that there is abolishment of soluble ST2 in the serum, but the normal ST2 is not changed in these knockout mice. And we were very excited to note that knocking out soluble ST2 in the AD transgenic mice, we observed the reduction in the A-beta plaque load. And very recently, we also saw a decrease in the serum neurofilament light chain. So even neurodegeneration can be partially rescued in these knockout mice. So we believe that soluble ST2 is a promising target for the therapeutic development for AD. And we have ongoing efforts to develop antibodies that can suppress soluble ST2 expression, antisense oligo, and we have screened more than 300,000 small molecules, and we have identified molecules that can suppress soluble ST2 expression. So in this talk, I have shared with you using IL-33 ST2 signaling pathway as an example, where I show you that IL-33 can stimulate the state transition of microglia from homeostatic to chemotactic to phagocytic states, and in so doing, enhance the clearance of A-beta. And I have shown you the detailed mechanism how the IL-33 signaling pathway can drive this kind of microglial state transition process, the involvement of VCAM1, recognizing ApoE, which is associated with the A-beta plaque. I have also shown you that we identify a genetic variant, a protective genetic variant of soluble ST2, providing the genetic evidence for that using the database of the Chinese population. And this particular genetic variant can influence the expression of soluble ST2 and confer a protective effect on AD risk. And leveraging the different model systems, including the cellular and the mouse model systems, as well as human data, we were able to dissect the pathological mechanisms of soluble ST2 in AD. And I believe that studies using the blood proteome would really help us to understand the biology of AD and also the genetic risk factors. I've also shown you the data using soluble ST2 as a therapeutic target, whereby we can start to find ways to induce and sustain the beneficial microglial functional state. And I also believe that circulating blood proteins can be potential therapeutic targets. And in fact, the proteome profiling study that we have performed really reveal many of such candidates that we should look at as potential therapeutic targets for AD. So I want to thank the members of my research team, and I have tried to acknowledge them as I go along during this talk. And they are truly outstanding, and they work very hard to ensure that we can make contributions to understand the biology of AD. I also want to thank my collaborators listed here, in particular John Hardy and Henrik Sederberg. And Henrik really provided us with a lot of advice on the blood biomarker studies, and John Hardy with the genetic studies that we have performed, and David Hosman for the ApoE study, Colin Master for sharing the Australian cohort, etc. Many collaborators to thank. I also want to thank the organizations who have provided us with the patient cohorts for our study, and also the funding agencies that have supported my work throughout the last 3 decades. So again, thank you very much for your attention. Thank you.
What an informative talk. Thank you, Nancy. For those who don't know me, my name is Long Jun Wu. I'm from University of Texas Health Science Center at Houston, transitioning from Mayo Clinic. So I serve in SFN program committee, and I'm today's moderator. So due to the interest of time, so we will just ask one. I got a lot of questions already online, but just ask one the key question. The question is that such a wonderful story on our 33 story. So does our 33 reach clinical trial, and how it can target specifically to microglia, any side effect you can envision?
So when we obtain the data using the AD transgenic mice where IL-33 show a clear beneficial effect, we were very excited. So we wanted to push ahead and explore the possibility of using IL-33 as a therapeutic approach. But IL-33 actually has peripheral side effects. And it is not a desirable therapeutic candidate because when we administer IL-33, for example, in the mice, we use IP injection, and we can accomplish the beneficial effect. But we found that if we go ahead, the undesirable peripheral side effect would make it not workable because we will have to work with dosage, and it's a very narrow dosage. So we try that, but the preclinical stage, we realized that it is not optimal. So hence, for AD, IL-33 as a drug would not proceed. That's why we went ahead and then looked at SOLO-ST2 as an alternative target, because for SOLO-ST2, we can find ways to suppress SOLO-ST2, and the proof of concept that I show you using the SOLO-ST2 knockout mice while maintaining the level of normal ST2, we found that in those mice, actually there's no growth abnormalities and so on. So we believe that SOLO-ST2 would be a more viable therapeutic target, and we are pushing ahead to explore that possibility.
Yeah. Thank you very much, Nancy. Due to the time, we will finish the Q&A session. The recording will be available in 24 hours, and also it's accessible in next 30 days. With that, let's thank Nancy again for her excellent talk. And thank you for your attendance and active participation, and enjoy the rest of SFN. I will see you around. Thank you.